Bismillahirrahmanirrahim. By now, we have seen that the granulation tissue is the active repair phase designed for rapid healing and filling tissue gaps, while mature scar tissue represents the final product, a stable, mechanically strong but functionally limited replacement. To achieve the final product, the connective tissue in the wound area needs to be remodeled. In this video, we are going to discuss the remodeling of the connective tissue in the wound area. These are the references for the video. The remodeling of the connective tissue, rather the remodeling phase of the connective tissue, is driven by a dynamic balance between extracellular matrix synthesis and degradation. Once extracellular matrix protein like collagen are deposited during repair, they continue to be modified and remodeled to shape a mature scar. The key players for this activity is matrix metalloproteinases. These are zinc-dependent enzymes that degrade various extracellular matrix components. The sources of these MMPs are fibroblast, macrophages, neutrophils, synovial cells, and some epithelial cells. The various types of these matrix metalloproteinases are present. One is interstitial collagenases. They are MMP1, 2, and 3, and they cleave the fibrillar collagen, while gelatinases that are grouped as 2 and 9, MMP2 and 9, they degrade amorphous collagen and fibronectin. Stromylysins that are MMP3, 10, and 11, they break down proteoglycans, laminin, fibronectin, and amorphous collagen. Their production is regulated by cytokines, growth factor, and signaling molecules. These MMPs are secreted as an inactive zymogens that require activation by proteases such as plasmin. Once activated, they exert their function, or they can be inhibited by inhibitors of metalloproteinases or TIMs that quickly inhibit them, ensuring the MMP activity is confined to the site of injury and there is a balance maintained. There is a related family of these MMPs, that is ADAMs, a disintegrin and metalloproteinase. These ADAM enzymes are membrane bound and they cleave extracellular domain of cytokines and growth factor like TNF, TGF beta, and EGF family members. This help regulate cell communication and signaling during tissue remodeling. The fibroblasts are early responders to injury and they are the key player in the wound healing. These are collagen secreting cells that build and maintain the extracellular matrix, and they are actually activated by a biochemical environment that is rich in cytokines and growth factors such as platelet derived growth factor, fibroblast growth factor, and transforming growth factor beta. They are involved in all stages of repair. In the inflammatory phase of the repair, they are recruited to the injury site in response to the chemotactic signal, and they are activated to begin extracellular matrix. Then in the proliferative phase, they proliferate and migrate into the wound and synthesize collagen, fibronectin, and other extracellular matrix protein. In remodeling phase, they participate in extracellular matrix remodeling and scar formulation, and they regulate balance between extracellular matrix deposition and degradation. They are also capable to differentiate into myofibroblast. Under the influence of cytokines such as transforming growth factor beta, these fibroblasts differentiate into myofibroblast. The myofibroblasts have got actin stress fiber that contain smooth muscle actin alpha, and they have got contractile ability. They pull the wound edges together and aid in wound contraction. Bone marrow also contributes into the wound healing. Some of the bone marrow derived mononuclear cell can adopt a fibroblast-like phenotype. These mesenchymal stem cells and fibrocytes are examples. They migrate to the wound and transiently contribute to the repair. The fibrocytes are thought to have a role in fibrosis and scar formation, and despite their recruitment, these marrow-derived cells do not become permanent resident of the local connective tissue. The fibroblast and myofibroblast are central to healing, but also to they lead to pathological fibrosis when the extracellular production, matrix production and remodeling go unchecked. For example, in liver, lung, and kidney, it induces fibrosis. Then these cells are also therapeutic target because if you understand the processes that has driven the development of and differentiation of these cells, you can go towards the antifibrotic therapies in chronic inflammatory diseases. So this image is providing an electron microscopic view of chick embryo fibroblast, and this fibroblast F is embedded in a collagen-rich extracellular matrix environment. So these fibroblasts are elongated, spindle-shaped cells with this is the elongated spindle-shaped cell with a dark irregular nucleus. The cytoplasm has got these blackish dots. These are actually scattered ribosomes, and it is showing a rich granular endoplasmic reticulum. There is presence of these fibroblast extensions that are reaching out towards the surrounding matrix. The collagen fibers appear as cross-striated structure. In some areas you can see these are cut transversely, and they are depicted as these black dots, and longitudinally as wavy fibers, these wavy fibers that are lying parallelly. The electron dense matrix here, these are the collagen fiber, dense organized matrix formed around the fibroblast. It is not uniformly dense that reflect the dynamic extracellular matrix remodeling by the fibroblast. So this is clearly showing the interaction of a fibroblast with surrounding collagen matrix, highlighting the active role of fibroblast in collagen production and organization of extracellular matrix, and the close packing and orientation of the collagen fibers typical of developing or repairing tissue. The cell's secretory and synthetic profile is reflected by the presence of organelles like rough endoplasmic reticulum. This picture is also showing a fibroblast that has got a nucleus, and over here this prominent central nucleus has got a well-defined nucleolus that is typical of active or metabolically engaging cells, and there is active cytoplasmic reticulum also, and abundance of this rough endoplasmic reticulum that surround the nucleus, that is surrounding the nucleus, and is responsible for formation of intense protein synthesis, that is production of collagen and extracellular matrix by these fibroblasts. Okay, then there is presence of these bundles of the collagen that are depicted by B, and they are reflected by these black dots in cross section, and some are wavy fibers. These are blackish dots of cross sections of the fibrin of collagen, and some wavy fibers of the collagen that are longitudinally cut. So these are the newly synthesized and secreted collagen. Then there are fibropositors that are highlighted by these arrows, the fibropositors. These fibropositors are actually the extensions of the fibroblast cytoplasm that surround and guide the fibrils of the collagen. These are the specialized structures through which the fibroblast orchestrate the assembly and extrusion of the collagen fiber directly into the extracellular matrix. Some of these newly formed fibrils are seen enveloped by the fibroblast membrane and cytoplasm, indicating the extracellular assembly and export of the collagen. The biological implication of these are there is active extracellular matrix synthesis. There is central role of fibroblasts in the production of extracellular matrix, collagen, and other structural protein. They have got dynamic interaction with extracellular matrix, and they do not simply secrete the collagen, they actively organize the collagen through fibropositor, contributing to the precise extracellular matrix architecture that is critical for the tissue repair and integrity. Also, they are responsible for healing and tissue strength because this process is fundamental for wound healing, fibrosis, and remodeling of the tissue. If the fibroblast properly function, that will work as tissue regeneration, or production of the collagen after the injury. If there is dysregulation in this process, it will lead to fibrosis or pathological effect of fibrosis in the organs such as lung, liver, or heart. This set of images display a series of histologic and electron microscopy images of fibroblast and myofibroblast within a tissue sample, and these are stained. The electron immunohistochemistry stain that is used in this is smooth muscle actin or SMA, which highlight myofibroblast. All three magnification 400, 40 and 1000x are used to highlight the architecture as well as the individual cell. The fibroblasts are spindle-shaped cells that are lightly stained uh, cytoplasm, while the myofibroblasts are spindle-shaped also, but with a darker, more intense brownish cytoplasmic hue or color that is due to smooth muscle actin positivity. So they clearly differentiate the fibroblast and myofibroblast, the fibroblast and myofibroblast on the basis of immunohistochemistry. The electron microscopic picture, this picture is showing the fine cellular detail. There is dense bundles of actin filament that are highlighted by these arrows, and these are the hallmark for their contractile phenotype. The surrounding extracellular matrix and these black dots or collagen fibrils are also visible. These images are from the site of fibrosis or wound healing that show fibroblasts transform into myofibroblast to produce and remodel the extracellular matrix. The myofibroblasts contribute to wound contraction and scar formation. In chronic injury, their persistent activation can lead to fibrosis, pathologic fibrosis, and liver cirrhosis, uh, lung fibrosis, etc. So this composite of light microscopy, electron microscopy illustrates the abundance of myofibroblast in that area, the ultra-structural feature that is actin bundle for contractility, and their role in the tissue remodeling and repair. In this image, the differentiation of fibroblast into myofibroblast and the role of transforming growth factor beta 1 and the mechanical tension in this process is illustrated. So here you can see that there is a fibroblast, and this fibroblast is an elongated cell with a central nucleus that is surrounded by a network of fibronectin. These green threads, these are fibronectin, these green threads, these are fibronectin, and there is presence of cortical cytoplasmic actin, that is this orange area. So no prominent alpha smooth muscle actin is seen at this stage. Then, under the influence of mechanical tension, the proto-myofibroblasts are formed that is an intermediate stage between fibroblast and myofibroblast. So the mechanical tension and transforming growth factor beta 1 signaling stimulate the development of alpha smooth muscle actin containing stress fibers that are these pink lines. These stress fibers are formed under the influence of mechanical tension and transforming growth factor beta 1. So these pink lines are developed, and these are still connected to extracellular matrix by focal adhesion. These pink ovals, these are focal adhesions. These focal adhesions. Through this, they are connected to the extracellular matrix. Then there is formation of differentiated myofibroblast. This differentiated myofibroblast is formed under the same stimulus, and it is a fully developed myofibroblast with prominent alpha smooth muscle fiber across the cytoplasm. The focal adhesions over here, they anchor the stress fibers, actin stress fibers, to the extracellular matrix. These are the EDA fibronectin or the fibronectin that are present in the extracellular area, and they provide contractility. EDA fibronectin, that is orange one, and the fibronectin, that is green, highlighting the extracellular matrix component that is involved. Over here, this column is showing the transforming growth factor beta activation pathway. Here, the latent transforming growth factor beta 1, it is produced by a cell and secreted and bound to latent transforming growth factor beta binding protein. And then the latent transforming growth factor binding protein with transforming beta 1 complex is formed, and this complex binds to extracellular matrix protein such as collagen, fibronectin, and fibrillin. Integrin receptors on the cell surface bind to this complex. The mechanical stress or tension that is exerted by the cell via actin stress fibers causes a conformational changes in the lab or latent actin latency associated protein, and this liberates active transforming growth factor beta 1, which can then promote differentiation of fibroblast and remodeling of extracellular matrix. So mechanical tension provides the stress fiber in the cytoskeleton of the cell, and it is essential for transition from fibroblast to proto-myofibroblast and then differentiated myofibroblast. The alpha smooth muscle actin and focal adhesions are critical for contractility and stabilization of the myofibroblast, and integrin in the focal adhesion link the actin cytoskeleton to the extracellular matrix proteins such as collagen and fibronectin. The fibronectin and EDA fibronectin provides a provisional matrix for fibroblast attachment and differentiation. So in this whole process, the transforming growth factor beta 1 is the central regulator of myofibroblast differentiation, and it promotes synthesis of smooth muscle actin, extracellular matrix protein like collagen, and fibronectin. So these myofibroblasts are essential for the contraction of the wound and scar formation. However, if they are persistent or pathologically active, they may lead to fibrosis in the vital organs. The transforming growth factor beta is the master regulator, promoting matrix deposition and suppressing the degradation. After an injury, the wound healing process involves the formation of granulation tissue that, as we have discussed, a transient vascularized tissue that is rich in cells and extracellular matrix component. One of the main players here is the fibroblast, a cell that synthesizes collagen and other extracellular matrix protein. So the early granulation tissue components have matrix that is composed of hyaluronic acid, proteoglycans, glycoproteins, and fine collagen fibers, primarily of type 3 collagen. These molecules provide a hydrated environment that promotes cell migration and angiogenesis. There is cytokine signaling. Uh, the damaged tissue release cytokines such as transforming growth factor beta, platelet-derived growth factor, and fibroblast growth factor. All of these increase the vascular permeability that lead to attraction of the inflammatory cell and vascular endothelial cell, a process that is called angiogenesis. Then there is activation of the fibroblast that occur 2 to 3 days after the injury. The morphology of the fibroblast changes from oval to bipolar or spindle shaped, and the collagen production is started. Now these fibroblasts start to produce type 3 collagen and fibronectin, and they also acquire contractile properties by transition to the proto-myofibroblast that express actin fibers for wound contraction. Initially, type 3 collagen is secreted in the repair process, and it is in large amount, that is fine reticular fiber for flexible scaffold for the tissue. As the healing progresses, type 1 collagen production overwhelms the type 3, and type 1 collagen forms thicker, stronger fibrils that restore the tensile strength. Final ratio of the collagen is about 80 to 85% is type 1, while only 15 to 20% is type 3 collagen in the later stages. So in 5 to 7 days of the injury, due to intense fibroblastic activity and matrix synthesis, uh, there is stabilization of the wound, and there is a mature scar form. Scar form type 1 formation, scar starts to mature, and in all these processes, transforming growth factor beta plays a critical role. It leads to increased synthesis of collagen, fibronectin, and tissue inhibitors of metalloproteinases. It decreases synthesis of matrix metalloproteinases that prevent the breakdown of extracellular matrix. Also, the wound maturation occurs by cross-linking of collagen. Collagen fiber, they undergo enzymatic cross-linking by the lysyl oxidase mediation, and this cross-linking increases the tensile strength. So the final outcome is going to be a stronger scar tissue that has got remodeled collagen with more type 1 as compared to the early cellular or granulation tissue. So all in this during all this process, the fibroblasts are central to the matrix accumulation during wound healing, while the type 3 collagen forms the early flexible scaffold, and type 1 replaces this type 3 and forms a dense, stronger matrix. And the master regulator of all this process is transforming growth factor beta that promotes the deposition of the matrix and suppress the degradation. The growth factors are important in the process of healing. The discovery of epidermal growth factor or EGF and many other growth factors has dramatically expanded the understanding of tissue repair and fibroplasia, that is the formation of fibrous tissue during healing. These growth factors are essential because they are important for interaction with the extracellular matrix component such as heparan sulfate, proteoglycan, matricellular protein, and decorin. They bind to growth factor and modulate their availability, enhance their effect by presenting them to cell surface receptor. They also provide biochemical context via integrin that link extracellular matrix with the cell interior signaling. Then there is concentrated localized signal. These growth factors don't act like circulating hormone. Instead, they work locally and persistently at the site of the wound. There are some therapeutic application and challenges for these growth factors, such as platelet-derived growth factor has shown to accelerate the healing in neuropathic diabetic foot ulcers, and some topical growth factor therapy has got limited success in complex or chronic wound because the target tissue may lack responsiveness, or diagnosis and wound specific factor may alter the outcome outcomes of this process. Engineered skin substitutes with multiple growth factor hold promise for improved healing of the large lesions, and they are also important in ending the repair process. While early signaling is well understood, how repair stops it remains unclear. There is reduced oxygen tension in later healing that triggers downregulation. Then there are CXCR3 receptor pathway that help to regress granulation tissue and limit scarring. Increased storage and decreased release of the growth factor may lock in the final matrix structural stabilization of the tissue. The final scar maturation occurs within weeks, and this is due to balance of collagen synthesis and degradation that stabilizes this process. However, the fibroblasts, they continue to remodel the scar that slowly alternates its appearance for years. So these growth factors are critical for early wound healing and assembly of the matrix, and uh, there is local regulation that ensure effective and controlled repair. They target multiple uh, processes together. One growth factor may target multiple processes within the healing process. So the balance of signal and matrix stability ultimately limit excessive scarring and convert granulation tissue into scar tissue. They drive cell migration and proliferation and also coordinate extracellular matrix deposition and remodeling, as we have earlier seen. There is balance in the healing process via both the activation and termination. More than 20 growth factors have been identified, such as epidermal growth factor, VEGF, FGF, PDGF, transforming growth factor, keratinocyte growth factor, and insulin-like growth factor, and they are usually redundant. Some, if the gene is deleted, some other growth factor may undertake the function of the absent growth factor in some conditions, because many processes and many growth factor take the action or take the role in the healing process. So there is selective expression of the receptor isotypes in different cell population. There is also temporal change in receptor presence, and there is also the zymogens or latent form of the growth factor that require activation. The cell surface receptors and proteoglycans and integrin, they help to localize and focus these signals and enhance the effect compared to the systemic hormones. In the early phases, that is inflammation, fibroplasia, and reepithelialization, growth factor like VEGF, PDGF, FGF, EGF, and keratinocyte growth factors are expressed, and they promote cell migration, recruit inflammatory and endothelial cell, stimulate fibroblast proliferation. They also initiate angiogenesis, while in the late phase, that is maturation and remodeling, the transforming growth factor beta and IGF-1, they are expressed, they play their part, and they promote remodeling of the matrix, stimulate the synthesis of the collagen and fibronectin production, and drive the transition from granulation tissue to the mature scar. This table, titled Growth Factor Control Various Stages in Repair, lays out the orchestration of repair signals, showing how multiple growth factors have overlapping and sequential role to ensure a well-regulated complete healing of the injured tissue. The main categories for the healing phases, such as attraction of the monocyte and macrophages, is done by platelet-derived growth factor, fibroblast growth factor, and transforming growth factor beta with macrophage chemotactic protein 1, that is C-type chemokine ligand 2. These molecules recruit monocyte and macrophages to the site of the injury, and crucial for the initiation of the inflammation and cleanup. Following this, there are many other specific chemoattractant or chemo or growth factors that are responsible for many processes, such as proliferation of the fibroblast, attraction of the fibroblast, angiogenesis, and others. For example, for the attraction of the fibroblast, the platelet-derived growth factor, fibroblast growth factor, transforming growth factor beta, connective tissue growth factor, epidermal growth factor, stromal cell drive growth factor 1 are responsible. Then for the proliferation of fibroblast, the platelet-derived growth factor, fibroblast growth factor, epidermal growth factor, the insulin-like growth factor, and connective tissue growth factor, they are responsible. For angiogenesis, VEGF, FGF, and hepatocyte growth factor, they are responsible for the collagen synthesis transforming growth factor beta, PDGF, insulin-like growth factor, and connective tissue growth factor are responsible. Collagen secretion. After the collagen secretion, the epithelial migration and proliferation, it is done by keratinocyte growth factor, transforming growth factor, hepatocyte growth factor, insulin-like growth factor of the epithelium and epidermis. Then for the resolution and healing, the gamma interferon inducible protein or CXC type chemokine ligand 10 and 11 or gamma interferon inducible protein 10. So IP10 and IP9 are responsible for resolution of the repair. In this table, it is shown that there is overlapping of the role by many growth factors such as PDGF, FGF, and transforming growth factor play multiple stage role. They drive both the recruitment and proliferation of the fibroblast, which are central to the tissue repair. They also support collagen synthesis and secretion, which form the extracellular matrix scaffolding. The VEGF and FGF, they are responsible majorly for the new blood vessel formation to nourish growing tissue, and the resolution requires IP9 and IP10. Epithelial growth factor like keratinocyte growth factor, transforming growth factor, they help to reepithelialization of the wound and restoring the barrier function of the skin. This table, growth factors, enzyme and other factors regulate progression of repair and fibrosis. It is providing an in-depth summary of the key growth factors, enzyme, and regulatory elements that are involved in wound healing and fibrosis, along with their effect at different stages. So the factors that are responsible for secretion of collagenases are platelet-derived growth factor, epidermal growth factor, interleukin-1, TNF proteases, and they promote the secretion of collagen degradation enzyme, that is collagenase. And then the movement of surface and stromal cells, this is regulated by tissue plasminogen activator, urokinase plasminogen activator, elastases, and various matrix metalloproteinases 1, 2, 3, 8, 9, 13, 14, and 19. These facilitate the cell migration and matrix remodeling by degradation of the extracellular matrix. Then there is maturation or stabilization of the blood vessel, that is by angiopoietin 1 and 2, platelet-derived growth factor, hypoxia-inducible factor 1, and they promote vascular stability and maturation. Then there is inhibition of the collagenase production, that is by transforming growth factor beta. There will be increased tissue inhibitor of metalloproteinases production, and they balance the activity of matrix metalloproteinases to prevent excess matrix degradation. Then there is a process of reduction in collagen production and turnover. There is reduction in mechanotransduction feedback and transforming growth factor beta release. Then the Due to this, the extracellular matrix is stabilized by reducing the turnover and activation of latent growth factor. Collagen cross-linking and maturation is done by lysyl oxidase, integrin receptor, fibronectin polymers, small proteoglycan, and this strengthen and mature the extracellular matrix by cross-linking collagen and organizing matrix component. So these MMPs, the family of matrix metalloproteinases, have got a highly specific role in breakdown of the matrix, such as collagen, gelatin, and stromatolysine. Angiogenesis and vascular stabilization is by angiopoietin, PDGF, hypoxia-inducible factor, and they help to mature and stabilize blood vessel during the processing of the healing. Transforming growth factor beta is the major regulator. It has got dual role. It inhibits the matrix degradation, the collagenases, and promote the extracellular matrix deposition. So the balance of extracellular matrix remodeling is by the activity of tissue inhibitor of metalloproteinases and matrix metalloproteinases, and stabilization is by lysyl oxidase and other matrix protein that increases the extracellular matrix durability. So the balance between MMP, TIMP, growth factors, and mechanotransduction, they ultimately determine whether repair leads to normal healing or pathologic fibrosis such as keloid formation or organ fibrosis. Thank you for watching this video. If you have any query, please write in the comment box.